Hello guys, welcome to my channel. So as you know, we are studying about the protein. So we studied about protein structure and we studied about protein function as well. So we are still studying about the protein function. So moving further into the topic. So as we discussed about the myoglobin, right? Or the hemoglobin. So that globin that we saw, there are like 4 kinds of globins that are found in humans, right? So hemoglobin, myoglobin, immunoglobulin, and cytoglobins. So now the protein structure affects how ligands bind. So we are studying about the ligands, right? So the binding of ligand to a protein is rarely very simple. So the interaction is greatly affected by the protein structure and is often accompanied by conformational changes. So what happens whenever the interaction is with the protein, ligand and protein interaction, the protein structure is affected and it's sometimes accompanied by the conformational changes to the protein structure. For example, the specificity with which the heme binds with its various ligands is altered when the heme is a component of myoglobin. So right, heme is not separate, like in the myoglobin, in the center of the myoglobin, heme is present, right? So whenever heme binds with its various ligands is altered when the heme is in the component of myoglobin. But for free heme molecules, carbon monoxide binds more than 20,000 better than O2, right? So in free heme, the affinity for carbon monoxide is 20,000 times better than oxygen, right? But it binds only 40 times better than the oxygen when the heme is bound in the myoglobin. So this is for free heme. But when the heme is bound in the myoglobin, right? So for myoglobin, right? Heme is in the center, right? So the affinity decreases to, so the carbon monoxide will bind 40 times better than oxygen. So that will be decreased. So for free heme, the tighter binding of carbon monoxide reflects the differences in the way the orbital structure of carbon monoxide and oxygen interact with the Fe2+, right? The heme has in the center Fe2+, that's the reason, right? It has in the center Fe2+. So the free heme, the tighter binding of the carbon monoxide reflects the differences in the way the orbital structures of carbon monoxide and oxygen interacts with Fe2+. So those same orbital structure lead to different binding geometries for carbon monoxide and oxygen when they are bound to heme, right? So as we studied, like the orbital structure is changed. So those same orbital structures that will lead to a different geometrics for carbon monoxide and oxygen when they are bound to heme. So I will show you. So for example, this is our Fa+ ion, right? It has the nitrogen and right, all of them. But when it binds with the oxygen, so it will be like this. So the geometry of that. And when it will bind with the carbon monoxide, right? Fa+. So, okay, so I need some space more. Alright, Fa+. It will bind like this, right? So this is a change in the geometrical of the oxygen and the carbon monoxide, and the change in a relative affinity for carbon monoxide and oxygen for heme when the heme is bound to a globin is mediated by the globin structure. So the affinity is mediated by the globin structure, right? So when the heme is bound in to the myoglobin that's inside the myoglobin, its affinity for oxygen is selectively increased by the presence of distal histidine in the myoglobin, right? So we saw that picture last time, and I will post it again, and you can see. So the oxygen affinity for the heme when it's bound to the myoglobin is increased by the presence of distal that's histidine group, right? So the Fe and Fe2 oxygen O2 complex is much more polar than the Fe2 carbon monoxide complex. So the Fe2 O2 is, this complex is much more polar than the Fe2 carbon monoxide. Why it is much more polar? Because there is a partial negative charge distributed across the oxygen atoms. So when this is attached to this, so there is a partial negative charge that is distributed across the oxygen atoms in the bond of the oxygen due to partial oxidation of this interacting ion atoms. And there is a hydrogen bond as well between imidazole side chain of histidine E7. So on your screen you will see the three-dimensional picture, and you can see the hydrogen bond that is developed between the imidazole side chain of histidine E7 and the bound oxygens, right, that stabilizes its polar complex electrostatically. So that is the reason the affinity of myoglobin for the oxygen is thus selectively increased by a factor of about 500. But there is no such effect for the Fe2 carbon monoxide binding in the myoglobin. So that's the reason consequently 20,000 fold stronger binding affinity of free heme for carbon monoxide compared to the oxygen declines to approximately 40-fold for heme embedded in myoglobin, and that is the reason this carbon monoxide, when its binding with the heme decreases, or rather it has just 40 times better than the oxygen instead of free heme when it's 20,000 times better than oxygen. Alright, and this favorable electrostatic effect of the oxygen binding in even more dramatic in some invertebrate hemoglobins. In hemoglobins where there are like two groups in the binding pocket, that can form strong hydrogen bonds with the oxygen, causing heme group to bind oxygen with greater affinity than carbon monoxide. So this selective enhancement of the oxygen affinity in the globins is physiologically important and helps prevent poisoning by the carbon monoxide generated from the heme catabolism or other sources. So this was the one factor. There is another factor, so that's also affect the binding of the oxygen to the heme in the myoglobin. So that depends on a molecular motions or breathing in the protein structure. So what does that mean? So that means the heme molecule, we know that the heme molecule is deeply buried in the folded polypeptide with the limited direct paths for oxygen to move from the surrounding solution to the ligand binding site, right? So if the protein were rigid, if you think like it's just a rigid protein structure, so the oxygen would not be readily enter or leave the heme pocket, right? So however, rapid molecular flexing of amino acid side chains produces transient cavities in the protein structure, and oxygen makes its way in and out by moving through these cavities. So there are like computer simulations that shows the pathways, but the most important was the distal histidine act as a gate to control the access to one major pocket near the heme ion. So the rotation of this histidine residue to open and close the pocket occurs on a almost like 10 to minus 9 nanoseconds time scale. So it's like something like this, nanosecond. So this is how much in the speed that it opens and closes the gate, right? So the distal histidine functions somewhat differently in some other globins as well, right? So like in neuroglobins or cytoglobins and some globins that are found in plants and invertebrates, the distal histidine is directly coordinated with the heme ion at the location where ligand must bind. So in these globins, the oxygen or other ligands must displace the distal histidine in the process of binding, and then with hydrogen bond again forming between the distal histidine and the oxygen after the binding occurs. Right, so that was myoglobin. Right, and now we'll see hemoglobin. Right, so hemoglobin transports oxygen, right, oxygen and blood, right? So nearly all the oxygen that is carried by the whole blood in animals is bound and transported by the hemoglobins in erythrocytes, right? So what are erythrocytes? That's red blood cells, right? So the normal erythrocytes are small. The normal red blood cells are small, and it's like 6 to 9 micrometer in diameter. It is biconcave in shape, right? Biconcave disc-like shape. Biconcave disc. So they are formed from a precursor stem cells called hemocytoblast. So these erythrocytes are formed from the precursor stem cells that are called hemocytoblast, and when it's in the maturation process, the stem cell produce daughter cells that form large amounts of hemoglobin and then lose their organelles, that is like nucleus, mitochondria and endoplasmic reticulum. So that is why erythrocytes are thus incomplete vestigial cells unable to reproduce, and in humans destined to survive for only about 120 days, right? But the main function is to carry hemoglobin, right, which is dissolved in the cytosol at a very high concentration, that is almost like 34% by weight. Right, so what the main function of erythrocyte is to carry hemoglobin that is dissolved in the cytosol. So in the arterial blood passing from the lungs through the heart to the peripheral tissues is about 96% saturated with oxygen. All right, so I will write it down. And arterial blood, right, arterial blood. So that is passing from the lungs through the, so it is passing from the lung to the heart to the peripheral tissue, right? Peripheral tissue. So the arterial blood, right, passing from the lungs to the heart to the peripheral tissues. In this, hemoglobin is about 6. In this, hemoglobin is about 96% saturated with oxygen. So this hemoglobin is almost carrying oxygen, right? But in the venous blood, right? So that is returning to the heart, right? That is returning to the heart. So in that, hemoglobin is only about 64% saturated with oxygen. So thus, each 100 ml of blood passing through a tissue releases 1/3 of the oxygen it carries, or 6.5 ml of oxygen gas at atmospheric pressure and body temperature, right? So every 100 ml of blood passing through a tissue releases 1/3 of the oxygen that it carries. Right, so the myoglobin that we studied is relatively insensitive to the small changes in the concentration of dissolved oxygen, and that is the reason it just functions well as an oxygen storage protein, while the hemoglobin, it has a multiple subunits and oxygen binding sites is better suited to the oxygen transport. So the interaction among the subunits in the hemoglobin can cause conformational changes that can alter the affinity of the protein for oxygen, and the modulation of oxygen binding allows the oxygen transport protein to respond to the changes in the oxygen demand by tissues. Right, and the hemoglobin subunits, so our structure is similar to the myoglobin, So hemoglobin is roughly spherical with a diameter of, right, so for the hemoglobin structure, it is roughly spherical and it has a diameter of about 5.5 nanometers, right? It has a tetrameric protein, right? So that contains 4 heme prostatic groups. Tetrameric proteins means that contains 4 heme prostatic groups. In each of the one associated with each polypeptide chains. So that has 4 polypeptides. So each associated with each polypeptide chains, right? So the adult hemoglobin contains 2 types of globin. So 2 alpha chains and 2 beta chain. So that is 146 residue each. Although the fewer than half of the amino acid residues are identical in the polypeptide sequence of the alpha and the beta subunit. So the adult hemoglobin contains 2 alpha chain and 2 beta chain, right? And the half of the amino acid residues for this are identical in the polypeptide sequences of the alpha and the beta subunits, and the 3-dimensional structures of the 2 types of subunits are very similar. So furthermore, the structures are very similar to that of the myoglobin as well, even though the amino acid sequence of the 3 polypeptides are identical at only 27 position. But still the structures are identical with the myoglobin. And all 3 polypeptides are members of globin family of proteins. Right, and the quaternary structure of this hemoglobin features strong interaction between unlike subunits. So for example, like alpha 1 beta 1 interface that's in the hemoglobin. So this alpha 1 beta 1 interface involves more than 30 residues, and its interaction is sufficiently strong that although the mild treatment of hemoglobin with urea tends to disassemble the tetramer of alpha beta dimers, but this dimer will remain intact, right? So the alpha 1 beta 2 interface involves 19 residues. So this alpha 1 beta 1 interface has 30 residues. Then there is alpha 1 beta 2 interface that has 19 residues, right? And the hydrophobic effect plays the major role in stabilizing these interfaces. But there are also many hydrogen bonds and a few ion pairs or the salt bridges, right, as well. Right, and one more thing, for the hemoglobins undergo structural changes on binding oxygen. So this is shown by the X-ray analysis. So when the X-ray analysis has been done, it has revealed 2 conformation of hemoglobin. So what kind of conformation? So the one is the R state and another is the T state. So this 2 state has been revealed by the X-ray analysis. So although the oxygen binds to the hemoglobin in either of the state, it has a significant higher affinity for hemoglobin in the R state. So the oxygen binding stabilizes the R state, right? So when the oxygen is bind in the R state, it stabilizes this state. So when oxygen is absent experimentally, it shows that the T is the most stable state and is thus predominant conformation of deoxyhemoglobin. So when there is oxygen is bound, so R state is more stable. When there is no oxygen bound, T state is more stable, right? So this T and R are just originally denoted as the tense and relaxed state, right, respectively, because the T state is stabilized by the greater number of ion pairs, right? When many of which lie in the alpha 1 beta 2 interface, right? So the binding of the oxygen to a hemoglobin subunit in the T state triggers a change in the conformation to the R state. So when the hemoglobin is in this T state and when T state, and when the oxygen binds to this T state, so it do a conformational changes to the R state. And when the entire protein undergoes this transition, the structure of individual subunit changes little, but the alpha beta subunits pair slide past each other and rotate. That means that will narrow the pocket between the beta subunits. Right? So you can see the picture on your screen. So that shows the transition from T state to R state. Now in this process, some of the ion pairs that stabilizes the T state are broken and some new ones are formed. Alright guys, so that was more for the protein function. So we are going through the protein function, but we are in the biochemistry as well. So that's why we are discussing the structural changes when the oxygen is bind to the hemoglobin or the myoglobin. Right, so here that does not finish this protein function though. So we'll still continue and we'll see you in the next video. So we'll discuss much more biochemistry for the protein function, right? All right. Thank you guys for watching and don't forget to subscribe the channel. Thank you.